complex biomolecules that are essential for all forms of life. Their sequences are encoded in DNA, which provides the blueprint for building these vital molecules. Proteins are made up of building blocks called amino acids. There are 20 standard amino acids that combine in different sequences to form proteins with unique structures and functions. These amino acids are linked together through chemical bonds called peptide bonds, forming long chains that fold into complex three-dimensional structures. Think of proteins as the workhorses of your cells. They perform a vast array of functions, including catalyzing chemical reactions, providing structural support, transporting molecules, and regulating biological processes. Proteins are built from 20 standard amino acids that combine in different sequences. Each amino acid has a common core structure consisting of a central carbon atom bonded to an amino group, a carboxyl group, a hydrogen atom, and a unique side chain called the R group. These 20 amino acids are the building blocks of all proteins. What makes each amino acid unique is its R group, the side chain that varies in size, shape, and chemical properties. The R group determines how the amino acid behaves, affecting protein folding and interactions with other molecules. Amino acids are categorized by their R group properties: hydrophobic, polar, acidic, or basic. For example, some R groups carry positive charges, allowing proteins to interact with negatively charged molecules. Amino acids are linked together through peptide bonds to form polypeptide chains. A peptide bond forms when the carboxyl group of one amino acid reacts with the amino group of another amino acid, releasing a water molecule in the process. This chemical reaction creates a covalent bond between the carbon atom of the carboxyl group and the nitrogen atom of the amino group, forming the peptide linkage that holds amino acids together. As more amino acids are added through repeated peptide bond formations, a polypeptide chain is built. The chain has a specific sequence determined by the order of amino acids, which forms the primary structure of the protein. The polypeptide chain extends from an amino terminus at one end to a carboxy terminus at the other, establishing the foundation for the protein's three-dimensional structure and function. The primary structure of a protein is simply the specific sequence of amino acids that make up the polypeptide chain. Each amino acid is connected by peptide bonds in a precise order. This sequence is completely determined by the genetic code encoded in your DNA. The DNA provides the blueprint that specifies which amino acids to use and in what order. The genetic code uses codons, three-letter sequences of nucleotides in mRNA, to specify each amino acid. For example, the codons UU and UUC both code for the amino acid phenylalanine. As the polypeptide chain grows, it begins to fold into regular repeating patterns called secondary structures. These include alpha helices and beta sheets, stabilized by hydrogen bonds between atoms in the peptide backbone. The alpha helix is a coiled structure where the polypeptide chain twists into a spiral. Hydrogen bonds form between every fourth amino acid, creating a stable right-handed coil. Beta sheets form when polypeptide strands align side by side, connected by hydrogen bonds between adjacent strands. This creates a pleated sheet structure that can be either parallel or antiparallel. Beta sheets often appear in proteins that need strength and flexibility, with hydrogen bonds creating the characteristic pleated pattern. Both structures are maintained by hydrogen bonds in the PED backbone, allowing proteins to adopt these stable conformations essential for their function. The repeating patterns of alpha helices and beta sheets provide the foundation for more complex protein folds. Hydrogen bonds between backbone atoms stabilize these secondary structures, with each bond contributing to the overall protein stability. The tertiary structure represents the overall three-dimensional shape of a protein formed through folding of the polypeptide chain. This structure is stabilized by various interactions between the side chains, or R groups, of amino acids. Key interactions include disulfide bridges between cysteine residues, ionic bonds forming salt bridges between charged amino acids, and hydrogen bonds between polar groups. Hydrophobic interactions play a crucial role in tertiary structure formation. In an aqueous environment, nonpolar amino acids tend to cluster together in the protein's interior, away from water molecules. Polar and charged amino acids, which are hydrophilic, position themselves on the protein's surface to interact with the surrounding water. This arrangement minimizes the energy of the system. The tertiary structure is maintained by a combination of forces, including hydrogen bonds, ionic interactions, disulfide bridges, and hydrophobic effects. These interactions work together to create a stable, functional protein conformation. The specific arrangement of these interactions determines the unique three-dimensional structure that enables each protein to perform its biological function. Some proteins are composed of multiple polypeptide chains or subunits that come together to form a functional protein complex. This arrangement is known as the quaternary structure. This diagram shows how individual protein subunits with binding sites come together to form larger complexes, such as dimers, helices, or rings. A classic example is hemoglobin, the oxygen-carrying protein in blood. It consists of 4 polypeptide subunits, 2 alpha chains and 2 beta chains, that assemble to form the functional quaternary structure. Another example is antibodies, which are composed of multiple polypeptide chains. They have 2 heavy chains and 2 light chains that form the Fab regions for antigen binding and the Fc region for immune system interactions. Proteins can be broadly classified into 2 main categories based on their shape and function: fibrous and globular proteins. Fibrous proteins are typically long, thread-like structures that provide structural support. For example, collagen is a fibrous protein that forms strong fibers in connective tissues like tendons and skin. In contrast, globular proteins are compact and roughly spherical in shape. They are designed for specific biological functions rather than structural support. Hemoglobin, for instance, is a globular protein that binds to oxygen in red blood cells and transports it throughout the body. The key difference lies in their structure and purpose. Fibrous proteins create the framework of cells and tissues, while globular proteins carry out dynamic roles in metabolism, transport, and regulation. This classification helps us understand how proteins contribute to the diverse functions of living organisms. Proteins maintain their complex three-dimensional structures through various intermolecular forces. These forces work together to stabilize the folded shape that determines protein function. Hydrogen bonds are one of the strongest intermolecular forces in proteins. They form between a hydrogen atom attached to an electronegative atom like oxygen or nitrogen and another electronegative atom nearby. In protein structures, hydrogen bonds help stabilize secondary structures such as alpha helices and beta sheets. Hydrophobic interactions occur when nonpolar amino acid side chains cluster together in the protein's interior, away from surrounding water molecules. These interactions are driven by the tendency of water to exclude nonpolar substances, effectively pushing hydrophobic regions together. This force plays a major role in driving protein folding and maintaining the tertiary structure. Van der Waals forces are weak attractions that occur between atoms due to temporary fluctuations in electron distribution, creating temporary dipoles. Although individually weak, these forces become significant when many atoms are in close proximity. In proteins, they help stabilize interactions between amino acid side chains and contribute to the overall structural integrity. Beyond the main forces, disulfide bonds and ionic interactions play important roles. Disulfide bonds are strong covalent links formed between cysteine amino acids, often stabilizing protein structure in extracellular environments. Ionic bonds, or electrostatic interactions, form between oppositely charged amino acid side chains, adding another layer of structural stability. These various intermolecular forces work together in a delicate balance to determine how proteins fold into their functional three-dimensional structures. Understanding these forces is essential for appreciating protein stability and function. Even a single amino acid change in a protein sequence can alter its structure and function. In sickle cell anemia, glutamic acid at position 6 of the hemoglobin beta chain is replaced by valine. This small change disrupts the protein's folding, causing red blood cells to sickle and block blood vessels. Hydrolysis is the chemical process that breaks peptide bonds using water, leading to the degradation of protein structure. Proteins consist of amino acids linked by peptide bonds. During hydrolysis, a water molecule targets these bonds. The peptide bond breaks, splitting the protein into smaller peptides or individual amino acids. This process is illustrated by showing how water breaks down a polypeptide chain. Hydrolysis can break proteins into peptides and free amino acids, as seen in this breakdown diagram. The process can be accelerated by enzymes such as proteases or by harsh chemical conditions like acid or heat. Denaturation is the unfolding of a protein, disrupting its higher-order structures such as secondary, tertiary, and quaternary structures. This process can be caused by several factors, including heat, changes in pH, or exposure to chemical agents. Denaturation often results in the protein becoming non-functional, as its shape is crucial for its role in biological processes. For example, heat can disrupt the bonds that maintain protein structure, leading to unfolding as shown in this illustration. Similarly, extreme pH conditions can alter protein charge and solubility, causing denaturation. Chemical agents can also break the bonds that hold protein structures together. In everyday examples, cooking or whipping can denature proteins in food, changing their texture and properties. The native folded state differs significantly from the denatured unfolded ensemble, as proteins lose their specific three-dimensional shape. For enzymes, denaturation at high temperatures prevents substrate binding, stopping the reaction. A protein's specific three-dimensional shape is absolutely critical for its function. This unique shape allows the protein to bind to specific molecules and perform its designated task within the cell. Think of it like a lock and key mechanism. In real proteins, this lock and key relationship is essential for biological processes. A protein's shape creates specific binding sites that only certain molecules can fit into, much like how a key only works with its matching lock. This precise molecular recognition drives all protein functions. When the right molecule binds to a protein, it triggers the protein's function. Enzymes use this shape-based recognition to catalyze chemical reactions, transport proteins carry specific cargo, and signaling proteins relay messages throughout the cell. The importance of a protein's three-dimensional shape cannot be overstated. It is the foundation of all protein function in biology. Even small changes to this shape can disrupt the protein's ability to perform its role, leading to disease or malfunction. Collagen is a fibrous protein that forms the structural framework of connective tissues throughout the body. Found in tendons, skin, ligaments, and bones, collagen provides tensile strength and elasticity. Its structure consists of 3 polypeptide chains that twist together to form a tight triple helix. The triple helix is stabilized by hydrogen bonds and features a repeating sequence of glycine, proline, hydroxyproline amino acids. This unique arrangement makes collagen incredibly strong yet flexible. In contrast, hemoglobin is a globular protein specialized for oxygen transport in the bloodstream. Each red blood cell contains about 250 million hemoglobin molecules. The protein consists of 4 polypeptide subunits, 2 alpha chains, and 2 beta chains, arranged in a quaternary structure. Each subunit contains a heme group with an iron atom that can bind 1 oxygen molecule. When all 4 heme groups are loaded with oxygen in the lungs, the hemoglobin changes shape slightly, making it easier to release oxygen to tissues that need it. Collagen and hemoglobin exemplify how different protein structures enable diverse biological functions. Collagen's elongated fibrous form provides tensile strength to tissues, while hemoglobin's compact globular structure allows efficient oxygen binding and release. Recent breakthroughs in artificial intelligence, particularly AlphaFold, have revolutionized protein research by predicting three-dimensional structures from amino acid sequences with near-experimental accuracy. This technology has already led to promising malaria vaccine developments by revealing the structures of parasite proteins that were previously unknown. These advances are opening new possibilities for drug design and understanding diseases at the molecular level.